So today I'd like to share with you some of our research work on identifying the role of epigenetic programming and reprogramming in anti-tumor or antiviral T-cell immune responses in order to boost cancer immunotherapy. So this is just a quick outline for our talk today. Oh, sorry. And basically, first we're going to show, or I'm going to actually share with you some of our published work identifying or showing the role of de novo epigenetic methylation programs in enforcing T-cell exhaustion, how full exhaustion of CD8 T cells represent a major challenge to cancer immunotherapy, and the potential of targeting these epigenetic programs in order to boost cancer immunotherapy. And then I will finally share with you some of our unpublished work showing some key microenvironmental signals and their impact on the human CD8 T cell exhaustion. In the last 2 decades, several advancements in our understanding of T cell immunobiology or immunology led to breakthrough discoveries for treating chronic virus infections or cancer. This was achieved by exploiting the selective killing power of one subset of our immune system called CD8 T cells or cytotoxic T lymphocytes. However, T cell exhaustion remains a major challenge to these T cell based immunotherapies. So before we talk about T cell exhaustion, first of all, what are CD8 T cells? So when you look at the lifestyle of CD8 T cells, you will be amazed. So we have in our blood a lot of naive CD8 T cells circulating. They are resting, they are quiescent, not performing major function. Once this naive CD8 T cell interacts or recognize the specific antigen presented on antigen-presenting cells like dendritic cells, they dramatically change their lifestyle from this naive, quiescent state into highly proliferating, highly cytotoxic state, generating what we call effector CD8 T cells or cytotoxic T lymphocytes. These cytotoxic T lymphocytes, their main job is to kill the virus-infected cells or tumor cells. If they succeed in their job and they clear the source of antigen, as during acute virus infections like flu or measles or even SARS-CoV-2 infection, most of these cytotoxic T lymphocytes die. Only a minor subset, about 5 to 10%, survive in your body, generating what we call functional memory CD8 T cells. These memory CD8 T cells can survive in your body for several years, or even decades, or maybe even lifelong longevity in your, or actually lifelong survival in your body. So this is the basis for the protection that we generate after vaccination or after specific virus infections like measles. These functional memory CD8 T cells, they have unique features. They can self-renew in your body without any exposure to this antigen. And another very important cardinal feature of memory T cells, they remain poised to rapidly recall the effector function. So I'd like you to keep this word in your mind, poised. Poised means a state of readiness. These memory T cells, they remain also quiescent, similar to naive T cells in your body. However, once you get re-exposed to the same epitope, the same antigen, the same virus, they rapidly turn on their effector function and go and become cytotoxic T lymphocytes. So this is a very unique feature in memory T cells. They maintain this poised state of rapid recall effector function. They have also different migration and tissue homing capacity. So some memory T cells reside in your mucosal layers, some reside in your spleen, others reside in your bone marrow. On the other hand, if these cytotoxic T lymphocytes fail in eradicating the source of antigen, and this occurs during chronic virus infections or cancer, examples for chronic virus infections like HCV, HIV, or HBV, what happens, these cytotoxic T lymphocytes progressively lose their effector function, generating what we call exhausted T cells. These exhausted T cells, they have poor effector function. They also have impaired homeostatic proliferation, which is different from memory T cells. And the other key feature on exhausted T cells, they maintain high expression of surface inhibitory receptors. Historically speaking, T-cell exhaustion was originally discovered by Wolfgang Nagel and Rafi Ahmed's lab back in 1998 using a mouse model of chronic virus infection using a strain of virus called lymphocytic choriomeningitis virus. The key point that I'd like to highlight from this cartoon, T-cell exhaustion is not black and white. It's not turn on, turn off. It's actually a progressive process. So at any point, you're gonna see heterogeneous populations in your body of exhausted T cells. Some cells are partially exhausted. They still retain the ability to produce some effector cytokines like interferon gamma or TNF alpha, while other cells are completely exhausted. And these fully exhausted T cells, they lost all their ability to produce any effector cytokines. Eventually, these fully exhausted T cells, they are physically deleted by undergoing apoptosis or cell death. Another key point that I'd like to highlight is, as I mentioned, exhausted T cells also upregulate these surface inhibitory receptors like PD-1 or CTLO4 or TIM3. These inhibitory receptors suppress T cell function. And this discovery, or actually these transcriptional findings, were the basis for the discovery of the first line of cancer immunotherapy that we call immunocheckpoint blockade therapy. The idea was to block these inhibitory receptors by using monoclonal antibodies that can bind to PD-1 or CTLO4. So these monoclonal antibodies were recently approved by the FDA to treat multiple types of previously untreatable cancers. And the list actually is expanding right now. I can tell you, if you check online, you're gonna see more than about 24 types of solid tumors and blood malignancy have been approved to be treated by this immunocheckpoint blockade therapy. Several types of these cancers are known to be refractory, not responsive to traditional cancer treatment, like chemotherapy, radiotherapy, surgery. For example, like metastatic melanoma, non-small cell lung carcinoma, or liver cancer and stomach cancer as well. This kind of immunotherapy made a revolution in cancer treatments. And as some of you know, Dr. Allison and Honjo, they were actually awarded Nobel Prize back in 2018 for their pioneer work in the discovery of these two receptors, CTLO4 and PD-1. Another line, or another new line of T-cell immunotherapy is called CAR T cells, or chimeric antigen receptor T cells. This T-cell therapy, it's kind of actually of personalized medicine. Basically, we take these T cells from a patient with cancer, and then we do genetic engineering or genetic modification of these patient-derived T cells by lentiviral transduction to make all these T cells express a receptor, which we call chimeric antigen receptor, that's specific to the tumor antigen. And then we expand these genetically modified T cells, and then we reinfuse back hundreds of millions of these CAR T cells back to the same patients. So this CAR T cell therapy showed very high efficacy for treating some types of blood malignancies, such as B-cell leukemia. However, it still remains ineffective for treating solid cancer or solid tumor. And one major problem for using CAR T cells in treating solid tumors is the development of T cell exhaustion. Despite this unprecedented success by immunocheckpoint blockade therapy for treating multiple types of cancer, there are still many challenges. A major challenge to immunocheckpoint blockade therapy is that many patients remain non-responsive to this immunotherapy. What we found in mouse studies and later in clinical research as well, immunocheckpoint blockade therapy can only rejuvenate partially exhausted T cells. It has limited efficacy against fully exhausted T cells. And you may actually think that most of those patients who do not respond, most of their anti-tumor T cells are fully exhausted. That's why they cannot respond to immunocheckpoint blockade therapy. So my question at that time was, what are the molecular barriers to immunocheckpoint blockade therapy? A key feature of T cells is their fate commitment and the longevity of their transcriptional programs. So we focused on a key mechanism that control transcriptional programs and feed commitment in T cells, which is called epigenetic programming. So what is epigenetic regulation, or what is epigenetic modification? So epigenetic modifications are stable covalent modifications in the chromatin that regulate transcriptional factor accessibility and gene expression programs without it changing DNA sequence. So this is not genetics, this is epigenetics, it's different. And one of the fundamental mechanisms that actually regulate epigenetic programming is called DNA methylation. We know that when you see DNA methylation within gene regulatory regions, such as promoter or enhancer, this is coupled with turning off gene expression. When you lose this methylation marks, this was shown to be coupled with either active transcription or turning on gene expression, or a poised state of gene expression. You remember when I talked about memory T cells? Memory T cells, they are not expressing their cytotoxic program. Once they get re-exposed or re-stimulated, they rapidly turn on their gene expression program. And this is because they are epigenetically poised. So their genes are ready to be turned on once you have the right stimulus. The nice thing to study, for studying DNA methylation, the machinery to acquire this epigenetic program is very well established. So for example, there is an enzyme called DNA methyltransferase 3A or DMT3A. This enzyme plays an important role in the acquisition of these new methylation programs. These programs can be inherited during cell proliferation, and this is mediated by another enzyme called DNMT1. So these programs are highly stable even during cell proliferation. So imagine you may acquire a certain epigenetic program, and even the daughter cells will maintain this epigenetic memory, although you already lost the initial trigger or the initial stimulus that regulated this epigenetic program. So there is something called heritability nature in this DNA methylation program. On the other hand, there are also another set of enzymes called TET123. These enzymes mediate hydroxylation of these methylation programs, which eventually cause demethylation of DNA methylation programs. So our hypothesis was DNA methylation programming enforces the functional impairment of exhausted T cells. To test this hypothesis, we need to experimentally develop T cell exhaustion in vivo. So as I mentioned earlier, one of the gold models to study T cell exhaustion is called LC-MEV infection model. The good thing about this infection or virus infection model, we have actually two strains. One strain causes acute infection. The other strain causes chronic infection in mice. Both strains induce the same type of epitope-specific CD8 T cell immune responses. So how can we assess the functionality versus exhaustion? Let me give you an example here by flow cytometry. And for those of you who are not familiar with flow cytometry, basically we have a cocktail of antibodies. These antibodies are specific to different markers on the immune cells, and each antibody is conjugated to different color, different fluorochrome. When you add these stained cells and run them through a machine called the flow cytometer, you can detect all the fluorescence colors or markers or signals from each individual cell. And then you can assess the expression value from each individual cell. So this is an example for the data here. This is called FACS plot. Each individual dot represents an individual cell, and the expression level in a two-dimensional pattern. You can see one example here like PD1, the inhibitory receptor, and this is GB33. This is a staining for the virus-specific CD8 T cells. And when you look in the spleen, 2 months after acute infection, where we should develop memory T cells, right? First of all, you see these memory T cells, virus-specific CD8 T cells, they have low expression of PD1. All right, once you do stimulation of these memory T cells by adding specific peptide, they rapidly turn on and express high levels of these effector cytokines, such as IL-2 and interferon gamma. In contrast, if you look at the splenocytes from chronic virus-infected mice, and this is 2 months after chronic infection, first of all, you see these exhausted T cells, they have high levels of PD-1, right? It's different from memory T cells. And also when you stimulate these exhausted T cells, you see this almost loss of expression of IL-2 and very limited ability to produce interferon gamma. And these two FACSA plots, you can use them as a guideline or actually as a textbook definition of T cell exhaustion. So in order to study the role of de novo DNA methylation programming in CD8 T cells, we used a mouse model to conditionally delete DMT3A enzyme inactivated CD8 T cells by crossing DMT3A flux and granzyme B clearly combinase mice. So the bottom line, we deleted DMT3A in CD8 T cells after they become activated. And then we gave them chronic virus infection. And we actually used a lifelong chronic virus infection, an extreme model of T cell exhaustion. So you can see by looking at the viral titers, both types of mice, wild type and knockout, they have these high levels of virus titers. However, when we looked at the virus-specific CD8 T cells in the blood of these mice, you can see at different time points post-infection. Initially, you have this good expansion, and then wild type CD8 T cells, the black line, they progressively lose their quantity and become exhausted. At this time point, within like 3 to 4 weeks, wild type CD8 T cells develop exhaustion. But when you look at DMT deficient CD8 T cells, they maintained significantly higher quantity compared to the wild type CD8 T cells. That was the first interesting finding. We also looked at CD8 T cells in lymphoid and non-lymphoid organs, and we have seen the same pattern or the same differences. What's more striking, when we examined the effector function of CD8 T cells in these knockout mice, we found that DMT deficient CD8 T cells, they can express striking amounts of these effector cytokines, interferon gamma and IL-2, while wild type exhausted CD8 T cells, they have this classic definition of T cell exhaustion. So this data suggesting that DMT3A is essential for the development of T cell exhaustion. So we went further and we identified what are these de novo DNA methylation programs that are acquired in exhausted T cells. We found that these programs were acquired in different genes regulating T cell trafficking and T cell effector function. This is just a list of genes if you are familiar with T cell immunology, but you can highlight one example here. Interferon gamma is one of these de novo methylation targets. And I just want to share with you kind of like example of how we look at this data. This is a snapshot from whole genome analysis, and each individual line represents an individual CPG site in the DNA. The red line represents methylation, the blue represents unmethylation, and the ratio between red and blue represents the percentage of methylation in this individual CPG site. So if you look at naive CD8 T cells in interferon gamma locus, it's heavily methylated. And this makes sense. Naive T cells do not express this effector cytokine. During the effector time point on day 8 post-infection, they lose these methylation marks and they become blue, as you see here, in both wild type and knockout mice. But during the chronic stage of the infection, when you look on day 35, wild type exhausted CD8 cells, they reacquire these red methylation programs, but the knockout do not acquire these methylation programs. So this is just an example how we identified these DNA methylation programs in exhausted T cells. So you can go back to this paper, but to just summarize, we found that during the development of T cell exhaustion, there is acquisition of specific and unique DNA methylation programs. These programs enforce the silencing of T cell function, and they also restrain the response to immune checkpoint blockade therapy. Building on this data, we thought to translate our findings into a therapeutic strategy in order to enhance the efficacy of cancer immunotherapy. And we asked it, what if we therapeutically erase these bad epigenetic programs in order to make these non-responsive T cells again responsive to immune checkpoint blockade therapy? So we used an FDA-approved drug called decitabine. This is a DNA demethylating, so it reduces the methylation level in a non-selective way. It's a DNA demethylating agent. So we used a very small dose of this treatment after the development of T cell exhaustion. followed by PD-1 blockade treatment. And we found synergistic expansion of the antivirus CD8 T cells across different organs in these sequentially treated mice, DEC plus PD-1 blockade treatment. So this data suggested that decitabine can enhance the rejuvenation potential of exhausted antiviral CD8 T cells. Similarly, we sought to test this new therapeutic strategy in a cancer model. So we give the same treatment regimen to a model where we injected prostate adenocarcinoma in mice. And we have also seen similar results. We found that sequentially treated mice, they had actually enhanced proliferation of tumor-infiltrating lymphocytes or TILs, CD8 T cells. And this was coupled with significant control of the tumor growth. So to summarize, we found that DNA methylation programs play an important role in enforcing T-cell exhaustion. These programs are highly stable, and they inhibit the response to immune checkpoint blockade therapy. If we target these T-cell intrinsic epigenetic programs, for example, by DNA demethylating agent, we can actually synergize the response to PD-1 blockade therapy. So moving forward, we had also collaboration with another lab in Germany, Dr. Dietmar Zen, and we found a unique DNA demethylation program in a transcription factor called TOX. So if you look here, TOX is heavily methylated in naive and in memory CD8 T cells, but it's completely unmethylated in exhausted CD8 T cells during chronic infection. This came out in Nature 2 years ago. In the same year, there are also like 4 other papers that came out supporting the major function that we identified for TOX in priming CD8 T cells for exhaustion. So you can see this is kind of like a downhill for naive T cells. It can go to either memory differentiation or to exhausted differentiation. TOX expression was a unique marker that enforced also, or that played an important role for enforcing this decision to go through the exhaustion pathway. So what are the implications for T-cell epigenetics in cancer immunotherapy? We may think about epigenetic reprogramming of T cells from this fully exhausted state back to the partially exhausted state. And that's what we try to do with this DNA demethylating agent, right? But the problem is these partially exhausted T cells can revert back to the fully exhausted state. Another better approach is to couple this reprogramming with diverting the effector T cells back or to the better pathway to generate functional memory T cells, which will provide us with a longevity or with a long-lived anti-tumor protective T cells. So my lab right now is focused on these 2 questions. In order to develop effective therapy to epigenetically reprogram these exhausted T cells, we need to understand and identify the upstream signals that regulate epigenetic programming in exhausted versus functional CD8 T cells. And then, based on these signals, we can actually develop T-cell targeted epigenetic therapy that can reverse this programming and drive T cells back to the functional memory state. So before I just share with you in the last few minutes some of our unpublished work, let me reflect with you on the signals leading to T-cell exhaustion. So this picture actually can reflect our state right now, right? And this is what we are experiencing, everybody of us, like what we call Zoom fatigue. And if you think about the main triggers for this Zoom fatigue, number 1, it's persistent self-evaluation, right? Especially for people who have to turn on their camera all the time. So you keep looking at yourself, right, in this camera. So this persistent self-evaluation, it actually induces kind of like persistent stimulation in your mind and distraction as well. The other thing, we are trapped in a box or a window, right? I mean, in our regular meetings, we can walk around, we can go here and there. The other or the third main trigger or problem that I think of is there is no coffee or cookies in our virtual meetings. That's exactly the same signals that you can think about that lead or drive T-cell exhaustion. So number 1, when T cells experience persistent TCR stimulation plus harsh microenvironment and also low support, low positive support, all of these key three signals drive or contribute to the development of T-cell exhaustion. So in order to identify what are the most important signals that we can use as a target for our therapeutic treatments, we actually did a robust integrative pathway enrichment analysis based on data sets derived from DNA methylation programs, chromatin accessibility, transcription. I don't want to bother you with all of these details, but the bottom line, we looked at the unique transcriptional and epigenetic programs in exhausted mouse or exhausted human CD8 cells. And then we found that TGF-beta is one of the most significant pathway that orchestrate these unique epigenetic and transcriptional programs in exhausted T cells. It's not the sole signal, but it's one of the top or the most significant pathway based on our analysis. So we thought to test this hypothesis or test this pathway in addition to others, and we actually developed a novel model to study human T-cell exhaustion in vitro. And the basic idea is to provide prolonged TCR stimulation in these human CD8 T cells in vitro plus cocktail of specific cytokines. And then we asked whether these TGF-beta or other signals will accelerate T-cell exhaustion. So we used T cells or PBMCs isolated from healthy human cord blood samples that have mostly naive CD8 T cells. And then we provided this stimulation for over like 4 weeks. And then we asked or we assessed the functionality and epigenetic programs in these chronically stimulated CD8 T cells. So this data, if you look at day 7 post-stimulation of human CD8 T cells and check their effector function, you can see that on day 7, these human CD8 T cells produce very high levels of this effector cytokine interferon gamma. So they are highly functional. But when you add TGF-beta from day 7 to day 14 and continuously provide TCR stimulation, you can see this huge reduction in the ability to produce interferon gamma. So plus interferon gamma, you see this significant reduction in interferon gamma frequency. And without TGF-beta, you still also see some reduction, but not as much as when you add TGF-beta. So this data suggesting that TGF-beta accelerates the exhaustion of human CD8 T cells. And this is just kind of like the summary of our analysis for the percentage of interferon gamma positive CD8 T cells in chronically stimulated CD8 T cells. As you see at all different time points, when you have TGF-beta exposure, this actually accelerated the exhaustion or the dysfunction of CD8 T cells by looking at the frequency of interferon gamma positive and also by looking at the expression level of interferon gamma per cell. The other thing that we asked, can we therapeutically reprogram this TGF-beta induced exhaustion in CD8 T cells? So we started using some of our cocktail of treatments. Number one, we used TGF-beta 1 blocker, and we have developed a new therapeutic strategy by adding another modulator for signal 3. So I'm just showing here a summary for our results. This is basically the interferon gamma expression level. From day 7, you have this high expression of interferon gamma. When you don't have TGF-beta and you keep stimulating T cells for 4 weeks, they still have this effector function. When you start adding TGF-beta, right, for another additional week, like this is just 1 week of TGF-beta, you see a little bit loss of effector function. 2 weeks, there is much more loss. 3 weeks, there is complete loss of the effector function. However, when you add this TGF-beta blocker plus signal 3 modulator, you see this huge regain of the effector function in human CD8 T cells. So this data suggested that therapeutic epigenetic reprogramming of exhausted T cells can be achieved by targeting TGF-beta and signal 3 molecules. And this is also another data showing similar results by looking at the expression level of T-BET. This is a key transcription factor in memory CD8 T cell function. And we found that this key transcription factor is progressively lost or reduced in exhausted CD8 T cells, especially when you add TGF-beta for 3 weeks. If you block TGF-beta and add the signal 3 modulator, you have this regain of the effector transcriptional program. This was also mirror image by looking at the DNA methylation programming in this T-BET or TBX21 locus, which expresses T-BET transcription factor. So as you can see, TGF-beta accelerated the acquisition of this DNA methylation program, but when you give this combined therapy, you lose this repressive DNA methylation program. So with that, I'd like to summarize with some of our current projects in the lab. Our ultimate goal is to induce epigenetic reprogramming of exhausted T cells back to the functional state. And this will be beneficial for treating cancer or chronic virus infections, right? We need, again, antivirus and anti-tumor T cell function. Understanding this programming or pathway will also help us for treating or for developing novel therapies to treat autoimmune disease. Basically, in some autoimmune diseases like diabetes, the major trigger for this autoimmune disease is the autoreactive CD8 T cells, which are highly functional. So if you can develop a strategy to induce exhaustion in these autoreactive CD8 T cells, you can actually block the progression of these autoimmune diseases. And in our lab, we use different models to study T cell immunology, like virus infections and mouse tumor models. And we also are trying to establish collaborations with clinicians here at OSU and outside in order to look at the human T cell epigenetic programs in CAR T cells or solid tumors, and also in chronic virus infection.